Our last speaker is Dr. Talia Moore. She is assistant professor in robotics and mechanical engineering from the University of Michigan. She's also affiliated with ecology and evolutionary biology and the Museum of Zoology at the University of Michigan. And you would think those two don't go together, but they definitely do. She uses biomechanical experiments to establish mechanisms for how biological structure determines the motion capabilities of animals. And specifically, she's going to talk about snakes today.
Hi, everyone. Thanks for watching this talk. I am really excited to talk to you about how and why to make a really convincing fake snake. So as some of us may know, the snakes are both predators and prey. They're not just out here eating other things, they are also being eaten themselves. And because of this, a lot of snakes are camouflaged, but some snakes tend to be pretty conspicuous, like these neotropical coral snakes. Now, this conspicuous coloration has evolved multiple times, at least 26 times independently across snakes. And some are venomous and some are harmless to most animals. So why do they have this color pattern? Well, we think that predators of these snakes recognize the color pattern and avoid snakes that they think are venomous. And these venomous and harmless species are often found in the same places, and this is what we would call Batesian mimicry. Now, the reason that we think these conspicuous snake color patterns experience less predation is because people in the past have used plasticine replicas. So on the left, you'll see these plasticine replicas that are painted to match real snakes. And if you leave them out in the world, you might find that the cryptic snakes get attacked more frequently than the mimetic snakes. But the problem is that every time this experiment is done, you've seen a little bit of variation. And what this means is that we actually also haven't seen a ton of features that we can use to predict the level of predation. So if we look in this Butch-Brody 1993 paper figure, it's not like the total amount of red, it's not the number of bands. We're not really sure what features can be used to predict the level of predation. And so I was attending another conference a few years ago, and I saw this talk on how colored organs might be moved to attract and retain gaze. So I thought maybe there's something going on with the snakes here. And yeah, there's actually been some research in snakes previously showing that snake movement and color pattern kind of interact to form an emergent anti-predator signal. So if you just look at color pattern or if you just look at movement, you're going to get kind of a misleading view of how this trait affects predation. And so I thought, what about these coral snakes? Do they have movement as well? And yeah, both the mimics and these venomous coral snakes do have these like thrashing movements. And this made me wonder, okay, how do color pattern and behavior interact to determine deterrence? And so instead of having kind of this like large three-dimensional type figure, I've turned this into a two-dimensional figure with heat map as this deterrence. So behaviors are on the left side, color patterns are on the top. And what I really want to do is measure the deterrent ability of each unique combination of color patterns and behaviors. And to do this, I want to make a soft snake robot that I can put out into the world, paint it different colors, have it move, and then record all of the predation events. But first, I need to make this, right? So to make this, I need to collect data. Let's look first at behavior data. Well, behavior data was previously characterized as binary traits, like yes, no, it does this or it doesn't. And that's for a few different reasons. Well, first, these snakes are super venomous, so you don't really want to hang out with them for too long. Second is that snakes behave pretty differently in the lab and in the field, so these really need to be observed in the rainforest, which is kind of hard to get to. And third, the rainforest destroys a lot of electronics, so it wasn't until recently when we've had much better waterproof camera technology that we've actually been able to take videos of these, because otherwise your equipment just fritzes in the rainforest. Another reason is because it was thought that these are signals that are like really important for deterring predation, and so we think that there's like strong stabilizing selection to reinforce that signal recognition. You want to be right near that peak Otherwise, you're probably going to get attacked. And so they didn't think that there was going to be much variation. So I decided to look into kind of the locomotion biomechanics of snakes. And the way that people have been studying the locomotion of snakes is often by attaching little markers to the snake that they can track through space. And that's not something that I really want to do with venomous coral snakes. The other thing is usually those snakes in these locomotion studies are going from point A to point B. So they're kind of physically constrained to always be moving forward. So, kind of the sum of all of the forces that they're generating on their substrate has to be pointing forward. So, they're never doing things like occluding themselves, which happens all the time in behaviors when you're not moving forward. I also looked into ethograms, like these behavioral ethograms are really cool because they can be used to measure and like kind of describe these behaviors through time, but there are some problems. So snake behaviors really differ greatly among species. And so an ethogram that is like custom made for one species of snake, like you see here on the right, is not necessarily going to have anything to do with the behaviors of another snake. Also, the body parts move independently. So if we look at the four bottom behaviors here, they're only differing in the movement of one part of the body. And so should we be considering these as distinct traits or distinct behaviors, or should we be looking at them as a little bit more related than they would be to, say, like the movements on top? Also, we found that in their literature, herpetologists often infer intent. So they might say, this snake is being defensive or it's exploring, and that requires like um, a lot of experience with those snakes to be able to infer that consistently. So we wanted to make it a little bit more objective. So I wanted to collect a taxonomically diverse data set of snake behaviors so that I could look at the variation in behaviors and really measure, how close are the mimics to these venomous models. So I wanted this to be context specific. I needed these behaviors to be captured in nature, and I wanted to handle them minimally. I wanted to get like a really high efficiency. So I wanted to integrate with ongoing field expeditions. I also wanted to do interspecific comparisons. So I used consistent capture methods, and I wanted these to be objective or as objective as possible. So I recorded all the behaviors on video. If I wanted to know where to go to collect a whole bunch of snake species all at the same time, my postdoc advisor, Allison Davis Roboski, actually published a paper looking at snake species richness globally. And so we used this paper to inform the location of our field sites. So we had 4 sites in Peru, we had sites in Nicaragua, Belize, and Panama. And this is what it looks like when you kind of go out into the rainforest on these expeditions. You're usually hiking for miles at a time every night, and you're carrying a big bucket with you. And this big plastic bucket is really important because if you catch a venomous snake, you put it in a cloth bag and then you seal it in the bucket so it doesn't bite you as you take it back to your field lab. So I thought this bucket is really useful. It's kind of standard, everyone has it. And I decided that I wanted to modify that bucket a little bit. So I cut a hole in the top and I put in a GoPro camera from the top. And then around the lip of that bucket, I soldered some Christmas lights, and I soldered them to be like pretty waterproof because that's really important when you're out in the rainforest. Now, a lot of these data were collected by these incredible students, Joanna Larson, who's now at Notre Dame, Erin Westing, who's now at Berkeley, and Sierra Sanchez Paredes, who's now at the University of York in the UK. And this is what it would look like when we would go out into the field. We'd find a snake, we'd put it in the bucket, and then we'd put the lid on the bucket and film. So these students went on 7 field expeditions. They collected 7,000 videos, almost 3 terabytes, and we collected 80 snake species and 367 individual snakes. And we also did another thing where I created this kind of pop-up behavioral arena so that I could go into these field sites where we had sort of these field labs that were pretty much part of the forest too. But we could isolate different types of stimuli. So vibration, looming, different kinds of sounds, and record their responses to these different stimuli. So here's a picture of me and Joanna and Xenodon severus. And then when I collected all of these video data, I had to find some way to analyze them. And so with the undergraduate research opportunity program here at the University of Michigan, I hired 16 undergraduate students to watch these videos and help me form a modular ethogram system. So they noticed that the head, body, and tail all have very distinct movements, and they can function pretty much independently from one another. And so we decided to create different modular ethograms for each body part. And so this is what the modular ethogram is like. In each body part, we have 5 different categories of behaviors. And then within each category, we have different mutually exclusive behavioral states. And so we have shape, presentation, position, posture, and movement for each body part. And you can go through every single frame of this high-speed video and just select, okay, it's going to be neutral, dorsal, on the substrate, neutral, and none. And you can go through and do that for the head, the body, and the tail for every single frame of data. So this is kind of what that looks like. I'll give you an example. This is a Xenodon severus responding to this looming behavior or looming stimulus. And this is what it might look like if you just take 3 different screenshots from that video. You might have, neutral, no gape, dorsal, partially off the substrate, neutral, none, and then you get a code for that. And so you can see how that code differs as the behaviors differ through time. What's really great is that this is really objective. And so these students, some of them who had never seen snakes before, were able to immediately get trained, and after about 2 weeks, start generating really, really great data. So this was one of the only research opportunities possible during the pandemic. Productivity actually increased, and I call this like a virtual expedition. So if you go into the Integrative and Comparative Biology journal from 2022, there is a paper on how virtual expeditions actually help support undergraduate research, especially during pandemics. The other thing is that all of these students are learning on the same set of videos. And so we looked at the variability in that first set of videos to compute scorer bias. And as you can see on this figure here on the right, if we take the same score for for many different videos, they're not very similar to each other on the left with these dotted lines. But then if we look at multiple different scores for the same video, they're going to be much more similar to each other. So far, we've got 477 videos digitized, and we're still going. So this is an ongoing research project. These time varying data are really, really helpful because we can start to think of them as almost like behavioral traits that might evolve, right? So we've got a molecular phylogeny on the right, and then we can start to build a behavioral phylogeny on the right, molecular phylogeny on the left. And we can look at these convergences and divergences and see that the behaviors really evolve with a different pattern than the DNA. So we're seeing a lot of convergence on these different behavioral phenotypes. The other nice thing is that we can do quantitative behavioral comparisons. So if we look at the coral snake, we can say, okay, these are the states in gape, presentation, position, posture, and movement that are most frequently found. And then we can take that pattern and compare it to a mimic and then to a non-mimic. And what we see is that the mimics and the coral snakes tend to share a lot of those behaviors, whereas the non-mimics don't share quite as many. Okay, great. So we've got these quantitative data on how snakes move. What do we do with them? Well, I want to make a snake robot, right? So I look in the literature for what snake robots exist. These coral snakes, they have fast thrashing, they have shape changes throughout their body, and I really want these things to be field compatible. So I don't want any electricity, I want it to be waterproof, and I want it to be autonomous. So I just want to leave it out in the world, go away for a month, and then come back and see what happens. I also wanted these to be safely huntable by predators so that when they bit into them, they wouldn't be electrocuted or they wouldn't be poisoned. So here are some snake robots that are out in the world now. This one on the bottom is a pneumatic snake robot, but it can't do these kind of thrashing motions. It kind of moves forward and that's it. But the nice thing about it is that it is kind of safely huntable and it's waterproof. Now, the one on the top can do slightly more complex behaviors, but it is quite slow. And it's electric and it's very, very heavy and it's not really autonomous. And so you would really need to be out there waterproofing it and putting it out into the jungle and maybe hiding in the bushes, but there's no guarantee that the predator is going to come eat it. So I happened to be working with some students who were working on pneumatic latex robots. And with these robots, they have an elastomeric enclosure, and then you wind a string around them. And that string is inextensible. So depending on the angle that you wind that string, it changes the behavior of that tube as it inflates. And so with PhD student Dan Bruder, who's starting actually as faculty this fall in mechanical engineering, we decided that we were going to make some pneumatic snake robots. And I tested them out on my dog, and it turns out that they are quite safe for animal interaction. So with a superstar undergraduate student, Maggie Kohler, I said, I've got these modular ethograms, and these are pretty much how a snake moves. And so I said, can you design something based on these ethograms? She said, yes, this is actually really great that you've made them modular like this, because she can do something like head triangulation and just isolate that behavior on its own. She can generate like a body sinusoid, which we do see in some of these animals. We've got a body irregular coil. And then we've got a tail coil, which we see frequently in the coral snakes. So this modularity turned out to be really key to allowing Maggie to create these different behaviors. And we see that modularity really does enhance functionality and manufacturability in other snake robots in the field, or continuum robots in the field. So that's really nice. We actually found that with a really small number of pieces, so just these 6 pieces, we can mix and match them, the head, body, and the tail, to match 3 different species of snake. But they look the same, sure, but can we quantitatively measure how similar they are to those real animals? So another student, Shannon Danforth, came up with a way to measure this quantitatively. So first we measured the duration of the thrashing, and it matched really closely. And then we looked at the curvature along the body. And if we see the length of the snake here on the bottom, the green is the real snake, the orange is the fake snake, and we see that the fake snake is well within the envelope of normal behaviors of the real snake. So we match it pretty closely. Okay, so we've got our behaviors. What about color patterns? We really need to, like, if we're going to make this snake robot, we need to figure out how to paint it, right? So when we went out and did our field expeditions, not only did we collect these behavioral data in this little pop-up field arena, but we also collected a bunch of color photos. So this is me in the corner kind of taking a calibration photo. And we ended up taking a bunch of these different photos of the dorsal and ventral sides of these snakes. And we noticed that there was a lot of variation. So not only was there variation in the snakes themselves, the lighting conditions in the field were really variable. Some days we had electricity, some days we didn't. Some days it was really, really cloudy. And sometimes we did this totally in the dark using just kind of lamps. We had a ton of variation in the size of the snake, and we had variation in the number of loops that we would use to wrap them and the different directions of wrapping. So this was kind of a really difficult problem to figure out, how do we isolate all of these different snake shapes from the background? So existing tools to do that really require landmarking. But landmarking, and this is so that you know what's important in your photo versus what's not important. So if you look at these butterfly wings here, you can look at this pattern in the middle, and then that's for the patternized toolbox. And then the ImageJ Mica toolbox, you can take a look at these different distinct regions of interest. But to do that, it requires landmarking, and landmarking is actually pretty labor intensive. So you need to identify homologous points that change shape between species. And you have to kind of do this all over every single photo, which is a lot of clicking. So this is what we did for the snakes. We had basically 2 curves, and we had 80 points on each side of the snake. And it took a long time to do this. So I had one undergraduate student who was willing to do this, and then she graduated and no one else wanted to do it. So we only had around 150 landmark photos. But this is where, like, being in a robotics department was really great, because I learned about neural networks as segmenting images. So if you have these images, like say you have the bus and you want to be able to isolate the bus from the background, you can train a neural network to do this. But the thing is, you need training data to make this happen. So I had a great computer science undergraduate student work on this with me. And what was really nice was we had a few of those photos that did have those landmarks, thanks to that one student who did the landmarking and then graduated. We also had these pre-trained weights from another data set. So then what we did was we randomly split up those photos and landmarks into a training pool and a validation pool. And then we subsampled from them, which we're calling tiles, to make a training set and a validation set. So what does that mean? We've got one photo. We randomly took a bunch of little samples, like these little tiles that are all sampled from the same photo. So this increases our data set size, and it also gives us like a lot more context, a lot of different context for what might be a snake and what might not be a snake. So we had 135 training pool photos, we had 16 validation pool photos, and then 32 tiles from each photo. And then we did our training and our validation steps, and this was our output. So we had really qualitatively high mask accuracy, but then I really wanted to be able to quantify this, right? So we wanted to know, okay, the number of pixels that were correctly identified divided by the number of pixels in the manually landmarked data. And so if we look at B, this is everything that's blue was identified as the background. And you can see kind of near the tip of the tail, it didn't really do a perfect job. And so this is where we see that discrepancy between the correctly identified pixels and the pixels that are in the landmarked data. Okay, so we've got this metric, and then we measured this on the dorsal, ventral, and dorsal and ventral data sets. So we trained just on dorsal and then validated on dorsal, ventral, and then dorsal plus ventral data sets. We did the same thing with ventral and the same thing with dorsal plus ventral. And it turned out that when we trained on the ventral data set, our accuracy got really low. And that's because the student that we hired to do the landmarking was such a good biologist that she was like, okay, here are the edges of these ventral scales, and then you can still see some lateral scales from this view. So she segmented right in between the ventral scales and the lateral scales, instead of segmenting kind of the edge of that snake. Whereas on the dorsal side, the edge does equal the limit of those dorsal scales. And so this is just something to be careful and keep an eye out for if this is something you want to do in the future. So then when we created this and when we got the output of that neural network to be as good as we wanted it to, it would give us these custom weights. And then we took those custom weights, and then once you have those, you never have to do training again. You can give it an infinite number of unlabeled photos and then do inference to get your masked photos. So when we did this, our larger data set, it was 60 times faster than manual landmarking, but there's really no limit to how much faster this can be. So if you have a larger data set, it's going to be even faster. But it's not 100% accurate, right? So I wanted to know, how accurate is it going to be? And is it going to be accurate enough for color analysis? So I looked at the output using this Mica toolbox from ImageJ. So the way this worked is I took the landmarked data from that one student, and then I looked at the learned outputs, right? And then I used the Mica toolbox to analyze the color patterns in there. And if you look at the color pattern energy with respect to red, green, and blue, the residual is quite low. And so we are really well matching, even though we don't have complete accuracy, it's not changing the nature of our results. So then we take those data and then we use them to paint our soft snake robots to resemble the captured snakes. So we have kind of a Lachesis muta, we've got an Oxyropus melanogenes, and we've got a Micrurus cerenamensis, and we can make these species-specific matches and then put them out into the jungle and find out what happens. So if you look down here at the bottom, you might see a couple of these little snake robots right here. And this is a photo from when we tried to put them out in the jungle right at the beginning of 2020. And then COVID happened. And so we all had to pack up and go home. So this is still in progress. We're still out here trying to look for the deterrence. We still want to collect these videos of predators trying to attack our snake robots. So I hope this, I really hope that this convinces you that it's important to think about how behaviors and color patterns might interact with each other, and not just think about these things in isolation, and that soft snake robots might be a really great way to collect this type of data. So thanks very much and have a great rest of the conference.